Thank you very much for that very kind introduction. I'm excited to be with you all this morning to kick off the week. So today I wanted to start by telling you a little bit about this new book that I just picked up from one of my favorite biographers, Arthur C. Benson. This biography is about Edward Fitzgerald, and he opens this biography with the following passage. So these are the first words of the biography. The life that is here proposed to depict was a life singularly devoid of incidents. Way to get everybody excited, right? It was the career of a lonely, secluded, fastidious, and affectionate man. It was a life not rich in results, not fruitful in example. It is the history of a few great friendships, much quiet benevolence, tender loyalty, wistful enjoyment. The tangible results are a single small volume of imperishable quality, some accomplished translations of no great literary importance, a little piece of delicate prose writing, and many beautiful letters. I don't think most biographers would start by bringing to mind so many disparate emotions, and I was sort of curious, what is it about this passage that I find so fascinating, and what would propel me to continue reading this biography? Is it the quiet benevolence and the tender loyalty and the loneliness and seclusion? Is it the fact that he had a really boring life, and I'm just fascinated by reading about a very boring life? Or is it the small volume of imperishable quality? In fact, the reason that I would continue reading this book is not because of any one of those pieces in seclusion, but it's actually the combination of them, that there was a boring life that led to one very interesting piece of literary contribution. Why and how? So what I'm interested in is, in fact, the network of interconnections between all of these different pieces. And that's actually no different than what many other people are looking for when they pick up a book. They're not looking for a single item. They're looking for how all of it fits together into an entire constellation. And the reason is that the knowledge that we are seeking when we're reading books or papers or web pages is a network. And Dewey, I think, puts this quite nicely in his 1916 Democracy in Education, where he says, knowledge is a perception of those connections of an object which determine its applicability in a given situation. Thus, we get at a new event indirectly instead of immediately by invention, ingenuity, and resourcefulness. And ideally, perfect knowledge would represent such a network of interconnections that any past experience would offer a point of advantage from which to get at the problem presented in a new experience. Now, as we move from book to book, or scientists as we move from paper to paper, or citizens as we move from web page to web page, we are each building knowledge networks. What sorts of networks do we tend to build? And do we build different kinds of networks? And those questions amount to the question of what is the curiosity that each of us has, and does it differ among us? To address this question, we have worked with a philosopher from American University, Perry Zurn, who performed a historico-philosophical examination of the use of words for curiosity in English, German, Latin, and French over the last 2 millennia. And what he did is that he created a taxonomy of types of curiosity that are present in literature and philosophy over those last 2,000 years. There are 2 kinesthetic signatures that I want to describe for you today. The first one is the hunter, who wishes they had a few hundred helpers and good, well-trained hounds that they could drive into the history of the human soul to round up their game in a targeted search for information. In contrast, the busybody, which is a second kinesthetic signature of curiosity, will frisk about and rove about at random wherever they please. Now, to test whether or not these 2 types of kinesthetic signatures of curiosity are present in humans today, we developed an ecologically valid experiment by having people browse Wikipedia, and this was work that was led by Dr. David Leyden-Staley. We operationalized the notion of these different kinds of curious productions using network science. So we hypothesized that this hunter would create knowledge networks that are very dense and highly clustered and with relatively short path lengths. And we hypothesized that the busybody would create these sort of much more circuitous paths with low clustering and long path length. Now, to test this hypothesis, we had individuals browse Wikipedia for 15 minutes a day for 21 days, which contributes over 5 hours of data per person. They visited collectively over 18,000 web pages. Now, to construct that, or to take that data and form it into a network, we had each web page be a node inside of the graph, and then we created an edge between two nodes in the graph using the similarity in semantics across the two different web pages. Now, what we observe is that there are massive individual differences in the types of curious search that individuals, humans, produce as they walk through Wikipedia. So on the bottom here, what you're seeing is differences in average clustering of the networks that are created and differences in the characteristic path length of the networks that are created. And on the x-axis, you see a measure of deprivation sensitivity, which is a measure of whether they seek to fill knowledge gaps inside of their mind. So what you see is a positive relationship between deprivation sensitivity and the average clustering coefficient. What that means is that the networks that when people have very high deprivation sensitivity, they walk through Wikipedia in a way that where sequential steps are two web pages that are very similar to one another. And in fact, they also come back very frequently to the same web page again and again. In contrast, someone who does not have very high deprivation sensitivity tends to walk through web pages in using much longer distance paths, so across very dissimilar web pages, and very rarely comes back to the same web page again. So this divergence allows us to see the behavior of both the busybody and the hunter inside of our personal browsing of Wikipedia web pages. Now, more generally, that suggests that individual differences in deprivation sensitivity leads to the creation of knowledge networks with distinct architectures. Now, we've been very interested in this observation because it begs the question, so when we seek information, it seems that we are seeking it differently, therefore constructing these different knowledge networks. Now, what happens when we go to learn a new field and that network has an architecture that differs from the architecture that we personally prefer to build? Do we then face learning difficulties? And just to give you an illustration of how that might happen, here are two different network architectures from two different fields of inquiry. So in general, science itself, I want to say, is actually composed of many different subfields, and each of those subfields can have very disparate network architectures. The fact that each of these fields has a network architecture that's characteristic of it is nicely pointed out by Henri Poincaré in his 1905 Science and Hypothesis, where he says, "The aim of science," and this is all of science, is not things themselves, as the dogmatists in their simplicity imagine, but the relations among things. Outside these relations, there is no reality knowable. Now, the two instances of these network architectures in two different fields are shown on the bottom, where I have words in a linear algebra textbook on the left, and then I have a network of dependencies between 20,000 mathematical proofs on the right-hand side. And what you can see just by eye is that there are striking differences in the network architectures of these two bodies of work. Now, when we are facing those types of areas, do our personal preferences in the networks we like to build constrain how easy it would be for us to learn these two areas of mathematics? To address that question, we constructed a highly stylized laboratory experiment to ask the question of how humans learn networks and how easy it is or hard it is for people to learn networks with different architectures. So we can define a network like the one that I am showing you here, and we'll let each stimulus be a word or an image or a movement, and that'll be a node inside of this graph. And then we'll let each edge in the graph indicate an allowable transition between two nodes. Then what we'll do is that we'll construct a sequence of stimuli by taking a walk through this graph and showing human participants each of those stimuli in succession, as defined by a walk on the graph. Then at each stimulus, we'll require the participant to perform a task. And the reason we do that is then their reaction time to the task reflects their expectations, and therefore how well that edge in the graph is learned. Now, if we look at the field of statistical learning and ask from that prior literature how humans would respond to the network on the left, our hypothesis would be that humans would respond equivalently. Their reaction times would be flat to every node on this graph because the transition probability on every edge is 25%. And the reason that is that if you carefully look at that node, either of the graphs here, every node has exactly 4 edges. And because every node has exactly 4 edges in both of these graphs, the transition probabilities on every edge is 25%. So if humans just care about transition probabilities, which is what we would think from a large body of previous work in statistical learning, then humans should respond equivalently to every edge. However, that's not what we find. And in fact, what we find is that humans are much faster at responding to nodes that are very deeply inside of modules, and I'm showing you that on the left-hand side in blue. And they're much slower in responding to nodes in the graph that are either at the boundaries between different modules or connected to a boundary between different modules. So what that indicates is that humans are not just sensitive to transition probabilities, but they're also sensitive to hierarchies in the networks that they learn. But that's not all. It's not just that they're sensitive to hierarchies in the networks that they learn, it's also that they are sensitive to the overall topology of the graph. So what you're seeing on the right-hand side is a random graph, again, where every node has exactly 4 edges, and humans are much slower in responding to a random graph than they are to a modular graph. So together, these two results reveal that humans process information beyond transition probabilities, being systematically sensitive to network topology. Why? So why do human expectations systematically diverge from reality? The reality is the transition probabilities are flat, and humans are not responding in that particular way. So why? We built a model of why human expectations systematically diverge from transition probabilities. It's dependent on the free energy principle, which suggests that humans want to minimize the computational complexity of their model and also minimize errors in response to their environment. And these two competing requirements actually enforce a local temporal integration of information that then explains these divergences from reality. So what that means is that we can then define the perceived information of any network. It could be the network of this book that I read earlier to you. It could be the network of the scientific paper that you read last night. And that perceived information is given by how complex the graph actually is, and that's the entropy that I'm showing you here, plus how divergent is that network topology from human expectations and human biases and expectations. Now what we can do is that we can plot, which I am doing on the right-hand side, how networks across different fields of our life, from language to semantic organization to web pages, citations, social networks, and music, how much information do they encode? That's the entropy, and what we can see here is that the entropy S is much higher in all of these graphs than expected in random counterparts. So there's a lot of complex information that's present in those graphs. And then how far do each of these diverge from human expectations? And we actually find that these network architectures diverge very little from human expectations and biases in human expectations. So that suggests that perhaps these systems have evolved, or these types of communication systems have evolved over long periods of time to process or to share, communicate as much information as possible while diverging as little as possible from human expectations. So this motivates several questions. Do humans preferentially create networks that we understand? All of those networks I just showed you are human created. So are we creating these networks that we understand? And perhaps if that's true, are non-human made networks made differently? Do networks evolve over time to more closely match human expectations, or vice versa? And perhaps what we all are most interested in here today, what happens in the mind when we learn networks? And this last question reminds me of a passage from Aristotle's Metaphysics where he said, "Mind thinks itself because it shares the nature of the object of thought, for it becomes an object of thought in coming into contact with and thinking about its objects, so that mind and object of thought are the same." So here we are, humans, mapping out the network architectures of the world around us. What does that do to our mind, and how does our mind meet those demands? Now, there's actually some really beautiful work from several other wonderful labs over the last couple of years that have demonstrated that neural representations of nodes inside of a network are more similar for nodes that are close together topologically in the network, and neural representations are farther apart for nodes that are topologically dissimilar, so farther away on the network. And this is work from Anna Shapiro and Matt Votvinnik in 2013, and Gevert and Behrens and colleagues in 2017 in eLife. So this motivates the interesting question, might the complexity of the representation differ by network context and change appreciably during learning? Now, to address that kind of question, we need to develop some methods, and specifically we need to develop a method that assesses representational complexity in the brain and its change over time. And so I'm just illustrating for you here on the bottom the idea that very early on in learning we can have one type of representation, and later on in learning we can have a different type of representation for the same stimulus. How do we map out the complexity of that representation and how it might change over time? To address this question, we used longitudinal imaging of humans as they learned the value of 12 different objects. So these are computer-generated objects you're seeing on the left-hand side. They each have a different value that's associated with them, from 1 to 12. And at any trial in the task, humans are shown two of these stimuli, and they're asked to say which one has the higher value. The actual value that's used at that point has a particular mean given by the $ I'm showing you, but also has a standard deviation of about 0.5, just so that there's some amount of variability. What you're seeing on the right-hand side is that humans learn the value of these objects over 4 different days, and on each day there are 3 scanning sessions. On the first day they have 396 trials, and that leads up to, at the end of the 4th day, 1,584 trials. So they're learning over a long period of time. At the very beginning of the experiment they have they're at chance roughly, and then at the end of the experiment they're closer to 95%. So each shape here is represented in the mind. We show them each shape separate from the learning. We show them each shape and ask them what the value is, and each shape therefore evokes a neural response across brain regions that contributes a data point in ROI space. Now, many representations across the task jointly form this point cloud, and the dimension of that we can actually quantify. The way that we quantify it is to assign binary labels denoted by color to the neural data, which is denoted by shapes. Now, if the data is organized in a relatively low dimensional space, then it will actually be very difficult to distinguish binary labels, and that's shown on the top row. However, if you map this in higher dimensional space, it'll be much easier to actually distinguish between the binary labels blue and red. Now, the average performance of this notion of separability over different possible binary assignments of the data gives you the separability dimension of those representations. Now here what I'm showing you is that the separability dimension correlates with the response accuracy across subjects. So individuals who have higher response accuracy, so perform better on this task, also have much higher separability dimension. That allows for easier distinguishability between stimuli associated with different values. Now, after that, what we can do is not just say that this is more separable, but ask the question about whether it's more or less separable than expected. So we compared these data to a null model that randomly reassigns the object association to the data, and that allows us to address how learners might balance the competing forces of minimizing resources to encode information while ensuring that the encoding is maximally informative relevant to the stimuli. So what you're seeing here on the right-hand side is the true data in red and then the null model in gold. I want you to notice the fact that the null model in gold has, on average, negative correlations. And what this means is that faster learners are actually using a lower embedding dimension on top of their high stimulus dimension. So what that means is that they actually have very efficient representations. It's not just that they can separate out and maximize the encoding relevant information, but they're also minimizing the amount of resources that they take to do that. So this allows for a particularly efficient representation of these stimuli. Now that we have a method to assess the complexity of representations and how that complexity and efficiency changes over time, we're still left with the question of how. What features of the brain might constrain the complexity of a representation and the degree to which that representation can change? And this last question reminds me another passage from Aristotle, this time from the Physics, where he says, "Now if there was a becoming of every changeable thing, it follows that before the motion in question, change must have taken place in that which is capable of being changed or of causing change had its becoming. So what is the change that is occurring before this alteration in representation, and what constrains how much change can actually happen? To address that question, I think we need to go back to the underlying network architecture of the human brain, which is obviously richly interconnected, and this is high-resolution diffusion imaging data using 720 diffusion directions using about an hour-long scan. Now, within that network architecture, structural links constrain the propagation of activity in a manner that is dependent on the link topology. And what that means is that understanding human brain network architecture could potentially provide some important clarity on the geometry of representations and on the degree and type of change that representations can display over the course of learning. But I don't think it's only going to help us to understand representations. I also think it's much more general than that, and understanding this underlying network architecture can help us to understand the strategies that the control strategies that the system can implement. All right, so to understand the constraining nature of network architecture, we want to understand how the propagation of signals in the network system depends on the pattern of links. Now, what we have empirically is shown on the left-hand side, a network of structural links empirically measured by neuroimaging, and we can use the highest resolution, best, longest scans possible to do that. But what we seek is more than that measurement. What we seek is a theory for how the change in activity in one region affects activity in other regions. Now, we've formalized that problem in the form of network control, where we say that the neural processes that the brain uses can be approximated by a model of signals traveling along white matter tracks. And we've been using a discrete time and time invariant network model of activity where the state of the brain x at time t+1 is equal to the structural adjacency matrix A, so that's a structural connectivity between all brain regions, times the state of the brain at time t, that's x of t, and then plus some control, which can be exogenous stimulation, it can be induced by external stimuli or a task, or it can be information that's presented from another region of the brain. And that's B sub kappa gives the number of regions that are controlled, and U sub kappa gives the control energy. Now, what do we do with this simple model to try to understand how structure constrains activity and states, and by extension, representations? Well, there are two types of control strategies that I think are really interesting to think about in the context of the brain and have been developed previously in the field of engineering, and they are average controllability, which intuitively is the ability to steer the system into many easily reachable states, and then second, modal controllability, which is the ability to steer the system into a few difficult to reach states. Now, those are the intuitions. Here is the underlying mathematics. So we have here at the top just the actual formulation that I just showed you of the model. So x of t+1 is equal to ax plus bu. And then underneath of that, I've written the controllability Gramian. Now, the controllability Gramian allows us to ask questions about not just what the structure of the adjacency matrix is, but how it would respond to perturbations given this model of dynamical flow. The average controllability of the system or of a region is given by the trace of the inverse of the Gramian. And then the modal controllability can actually just be derived from the information from the eigenvectors and eigenvalues of A. So if we let V sub j be the jth eigenvector of A with a particular eigenvalue lambda j, then if the element Vij is small, that means that the jth mode of the brain, or of this network system, is poorly controllable from node I. That allows us to define a general scaled measure of controllability over all n modes of the system, where n is the number of regions in the graph, from a particular region I. Now, in the first study where we used this model, we asked the question of whether there are particular parts of the brain that show preferences for using either average or modal controllability strategies. And what I'm showing you here on the left-hand side is the controllability statistic of the top controller nodes in the brain. Either average or modal is on the x-axis, and in blue, I'm highlighting regions of the default mode system, and in red, I'm highlighting regions in the frontal parietal cognitive control system. And what you can see is there's a clear preference for default mode regions to show high average controllability, and there's a strong preference for frontal parietal regions to show strong modal controllability. So what does that mean? Number one, it means that regions implicated in transient control processes to push the system into a difficult-to-reach states are high in modal controllability and therefore structurally predisposed to be able to enact this state transition really simply with very low energy. Secondly, regions that are active at rest in the default mode system show white matter connectivity patterns that are predicted to effectively drive the brain into all nearby states. And one could argue that that's a relatively optimal way of designing a system, to place it at rest in a position where the regions that are active can push you into most states that you need to go. Subsequent to that first study, we've also shown that different brain regions have more or less power to alter whole brain dynamics, that the capacity for brain regions to change dynamics grows as children develop, and this is a study that uses the Philadelphia neurodevelopmental cohort, children or youth from the ages of 8 to 22, and that's what I'm showing you here on the right-hand side. On the y-axis is modal controllability, on the x-axis is average controllability, and color indicates age. So what you see, number one, is that there's a positive relationship between these two types of control, where human brains that have low values of modal and average controllability occur at the same time, whereas brains that have high average controllability also have high modal controllability. Intuitively, what does that mean? It means that brains that can perform one kind of control can also perform the other kind of control. Now, individual differences in network controllability also correlate with false positive rate on a continuous performance task, which is a common measure of impulsivity. So together, these results suggest that our theory is a useful marker of how the brain enacts control to change state, and that in turn affects the potential for alterations in representations, if we think about representations as patterns of activity or states of the brain. Now, I want to make a note that these notions of average and modal controllability and the structural predispositions for control are, I think, important, but they do miss the fact that in the human brain, we have a particular state that the brain is in that then may move to another state. Average and modal controllability just sort of assess, can you move to similar states, can you move to different states, is a very general question. But we might want to ask the question about precise control of specific state transitions. So here is a schematic example of, let's say I have activation in motor cortex, and I want to ask how easy is it for the brain to transition to a state of activation in visual cortex? How much energy would be required from which regions? And that type of question is also interesting in the context of clinical populations, which I'll show in a few minutes. Is that state transition easier or harder for someone in a healthy state versus in, for example, a state of mental illness? So what we want is not just a question of controllability statistics in general, but we want a way of thinking about these specific state transitions. So what we do is that we develop a method for finite time, finite average, multi-point control, allowing initial and target states. Building on that same underlying model of dynamics, we can define a cost function which penalizes the energy and distance of the current state of the brain from a target state, and that's shown on the bottom. And this is work illustrating the application of that question to the activation of frontal parietal circuitry. This is work that was done in Ted Satterthwaite lab at the University of Pennsylvania in the Department of Psychiatry and by his postdoc, but I wanted to showcase it here. The question is, how can a brain move from a baseline state to a state of high activation of the frontal parietal system? And this data set that we use to ask this question is, again, the Philadelphia neurodevelopmental cohort, and we use 946 individuals from that group. And what we find is that the control energy that is required for this particular state transition, so activation of frontal parietal cortex, decreases with age. So as we go from 8 years to 22 years of age, the amount of energy that's required for that transition decreases. More importantly, I think even the control energy that is required for that transition is correlated with executive performance on a large cognitive battery. So this indicates that greater executive performance is associated with brains that require less control energy to enact this particular state transition. Interestingly, the control energy in rostral cingulate is also able to mediate the effect between age and executive function. So this gives an initial piece of evidence to suggest that the control energy that we can estimate from the underlying models is something that relates to executive performance and changes with age in a way that we would hypothesize to be true given alterations in executive function with age. Now, another extension of this idea is to ask, let's look at activation profiles from tasks that we often use in cognitive neuroscience, let's say the NBAC working memory task. So you can actually think about the beta weights from a GLM of task activation as a state of the brain. And you can ask, how does the brain move from this particular state of activity to this particular state? So this is work that was done by Urs Braun in collaboration with Heike Toast and Andreas Meyer-Lindenberg at the Central Institute of Mental Health in Mannheim, Germany. And what he studied is activation in a zero-back task, and then how that changes to activation in a two-back task. We can take these two initial and final states and estimate how much control energy was required for that state transition. What we see is that the control energy is required is much higher when we go from zero back to two back than when we go from two back to zero back. And that's consistent with the direction of cognitive demand that's necessary for those transitions. What we also can do is to ask whether a particular state of the brain is easy to hold steady. So if we're in the zero back condition, can we stay in zero back with relatively low energy? Or if we're in the two back state, can we stay there with relatively low energy? Now the way we ask this question is to say, let's let the initial state be zero back and the final state be zero back, and then estimate how much energy is required for that. Or is the initial state two back and the final state two back, how much energy is required for that? That allows us to assess the stability of these states energetically on top of the underlying white matter. And that's what I'm showing you on the far right. So the stability of zero back is significantly higher than the stability of two back, which is again consistent with the differences in the cognitive demand of these two states. Interestingly, also, that stability of the two-back state is associated with individual differences in accuracy. Now, we also observe that there are deficits in this control energy in schizophrenia, particularly as these individuals transition from the zero back to the two back task. So we know that schizophrenia patients have deficits in executive function, and they have widespread alterations in structural connectivity. So we ask the question of whether they need more control energy for these state transitions, and whether that could offer an explanation for why we see changes in their performance. What we observe is that the control energy that's required for a schizophrenic or a patient with schizophrenia in this cohort to go from 0 to 2 is higher than it is in healthy controls. Interestingly, we do not see a difference in the control energy required to move from 2 back down to 0 back. So this suggests that this control energy theory could offer an explanation for changes in performance and the difficulty of the task for these patients. Now, hopefully, you're all wondering, what is it about a network that allows it to enact these state transitions in a relatively low energy way? If it differs in people with schizophrenia, obviously that's important. If it differs across development, obviously that's important, but I haven't shown you why. What is it about the network that's different? And to address that question, we could perform numerical experiments to say, what is it about a particular network architecture that predicts changes in control energy, and that would be important. However, I think even more important is to go back to the underlying mathematics and say, what is it, independent of any numerical simulation, about the network architecture that facilitates or hampers control? So Jason Kim in the lab worked on this problem, which was just recently published last year. And to do analytical work or mathematical work on network and network control is actually quite difficult. It turns out that if you take the entire network organization and try to understand it mathematically, that's hard, but some simplifying assumptions can be applied that help us to get a little traction. So one simplifying assumption is that we can just focus on the regions of the brain in which energy is injected, again, either from stimulation or from exogenous stimuli coming from a task. And then those are the red nodes here in the graph. And then which regions of the brain do we want to be able to change their activity? And those are regions that are colored in blue. So I'm going to call them the driver nodes. That's where the energy is injected. And the non-driver nodes, that's the ones where energy will flow to. So if we take the entire network and just focus down on this bipartite graph between where we inject energy and what we want to change, we can get a little bit farther. What I'm showing you on the bottom is a full Drosophila network. I'm going to show you a human in a little bit, which, and just is highlighting that when we look at that bipartite graph, it's just a tiny area in this matrix. Now, what we can do is that we can derive the energy that is required for these state transitions. And I have a equation here that I'm not going to explain all of, but what I want you to notice is that in the denominator of that fraction, you see the determinant of Q. Q here is this bipartite subgraph of just where energy goes in and where energy gets to. And that dependence on Q actually allows us to get in physical intuition for what it is about a network in different periods of youth versus adulthood or in health and disease that predict the energy that's required for control. And for that, what I have here is showing different instances of a graph. So there's a graph on the left-hand side, and then there are different instances of weights on those edges in the middle column, and in the final column is the determinant of Q, and that's what is in the denominator in that energy formalism. So what you see at the top is that when you have edge weights that maximize that volume of this parallelotope, which is the determinant, they're large in magnitude and orthogonal to one another. Whereas in the bottom, if you have edge weights that are very similarly distributed to one another, you get a very small determinant. So what that means, the big take home here, is that dissimilarly distributed weights on edges into these regions that you want to control their activity decreases the energy that is required for control. So in other words, complexity, dissimilarity in edge weights across the brain is extremely important for allowing low energy transitions. So what that also means by extension is that individual differences in this complexity or this dissimilarity of edge weights, and even regional differences in that dissimilarity of edge weights, constrain the states that the brain can easily move to, and by extension, the representations that the system can easily reach. Now, it's interesting to ask how we stack up in comparison to other species. What we show in the paper is that actually if you move from Drosophila to mouse up to human, that progression is increasingly favorable for control. So the topological changes allow for these transitions between diverse states in a way that is not something that we see in mouse or Drosophila. Again, also suggesting that the types of representations that humans could reach are quite different and more varied than other systems. So just to give you a pictorial representation of what we've done over the last couple minutes, we started off by asking the question of how we are curious and how we are different in our curiosity. And that led us into a discussion about network learning, which then led us into a discussion of perceived information in complex networks from language to music. Then we asked the question of how does the brain learn networks, and what is it that changes in the brain when humans learn networks? That led us to a question of developing methods for efficient neural representations of objects or items, and those are the nodes inside of the network. That doesn't necessarily tell us how people are learning the edges. That's for another day. And then from there, we asked the question of how these representations can occur easily or more difficultly, and how we can transition between representations. And that led us into the question of network control as a constraint on representations. We followed that by the question of energetics, the energetics of network control. Now, I want to make a last note about why did I spend half of the talk on non-brain work? And that was actually very purposeful, because I think that we as a community do really develop sophisticated methods to understand the human brain and to understand what each region of the brain does, if that's a good question, as Tal was asking earlier. But I think it's also important for us to consider human behavior and the complexity of human behavior with the same level of sophistication and the same level of interest as we do with the human brain, because that's really when we will understand what it is that the brain is doing. And with that, I'd like to acknowledge the people who are in the lab, the collaborators on this work, the funding agencies that have supported the work, and thank you all for listening.
So Dr. Bassett has indicated she'll take some questions. We only have 2 minutes, so if someone would like to ask a question. If not, I will start. Just in relation to the earlier work you presented about searching, how people search in websites. To what extent is that culturally dependent and also search engine dependent? Because the path that you take to get to various websites might vary.
Yeah, that's a really good question. We don't know the degree to which it's culturally dependent. I think that would be an interesting question to pursue in future work. It may depend to some degree on the websites in the sense that the hyperlink structure, if you'd use the hyperlink structure, will somewhat influence the URL passage. But in this particular experiment, they were also using just the search bar, so they could ask whatever next page they wanted to get to.
Okay.
So it's less important there.
There's a question here.
Yeah.
Yes, thank you for your talk. I've got a question about the connectome slide that you showed us just shortly before the end. Did I see it correctly that the scales were different, so that the scale of the Drosophila and of the mouse connectome was different from the human, and if you just put them on the same scale, the image would be pretty much the same?
That's a good question. Are you asking whether control energy depends on the scale of a system?
Yes.
Yes. So what we found is that the control energy for a system actually maintains some amount of consistency across different scales, although if you vary widely in scales over many orders of magnitude in n, then it can be different. But even if you hold the scale exactly the same, the driver seems to be the underlying topology of the two.
Okay, thank you.
Sure.
Let's thank Dr. Bassett for a very wonderful presentation.
Thank you.
There we go. Thank you. Okay, so the first award is for human brain mapping, and just a little bit of procedural. We invite all the associate editors of human brain mapping to nominate a paper, the best one from the year that they managed, if they found that one of them was exceptional. And so what we find is that about half the associate editors say, none of mine is good enough for the award, and then about half say, this was the best paper I managed. One thing that we've done for the past few years is all the nominees go open access on the Human Brain Mapping website. And so sometime today or tomorrow, anybody who's interested can go see if they were nominated for the Best Paper Award. I'll be sending out emails later, but all the nominees will go up, so they'll live soon. However, then those are turned over to the editors-in-chief, of which for human brain mapping there are 4, and we all read them and score them. And this year, this was the winner. So the editor-in-chief, or excuse me, the associate editor who nominated this was Paul Thompson. And so his comment in recommending this for the award is written there. I think it was quite a clever study. Tractography is very popular, but there is an increasing number of papers pointing out problems with various methods, including biases, which can make the whole area seem like a bit of a house of cards. This paper does a serious job of looking into some methodological flaws and points to be careful of, which I think makes it a very satisfying paper intellectually. The conclusions would not be obvious at all in my opinion, so I think the paper is very useful to the community. Specifically what they did was they compared DTI tractography with post-mortem anatomy and found that DTI tractography selectively tracked from gyral crowns and not from sulcal banks. Their observation was that when you do DTI tractography, you're biasing your result toward gyral crowns and omitting basically 2/3 of the cortex from the tractography. Fairly influential observation for those who use DTI tractography. The authors are listed. The first author, Kurt Schilling, is unfortunately not able to be here, and he has sent in his stead another member of the laboratory who will accept the award for him. And so Victoria Morgan from Vanderbilt University, please come forward and accept the award. Congratulations.
It's an honor to accept this award on behalf of Kurt Schilling and his group in the Vanderbilt University Institute of Imaging Science. And Kurt did this work during his PhD, getting his PhD work, and we were lucky enough to have him stay on at Vanderbilt for his postdoc, working with all of us, so I just hope that he does not find a bias in the methods that he uses with me. Thank you.
Okay, I'm Michael Breakspear, and I'm the Editor-in-Chief of NeuroImage, and I'm really thrilled to be here and announce the Paper of the Year award for NeuroImage. Papers are the beating heart of science, and so we take this award very seriously. As with Peter, we have an internal process. Papers are nominated by editors and the entire editorial board. They're selected from over 300 original submissions that we have every year at NeuroImage, and the resulting list is voted by the senior editorial team. The winner-up, the runner-up, sorry, I've got a terribly heavy head cold. I just want to take this opportunity to thank the authors, the reviewers, and the editorial team of NeuroImage, and welcome the new editors, in case you haven't noticed, who are listed here. Just to make an announcement, we're about to launch some new toolbox software papers. And as with our cousins at Human Brain Mapping, I reassure the community that we're moving with the times around open access developments. Okay, so highly commended, or the runner-up, is a beautiful paper from Zurich by Rike Peschner. And its focus of attention modulates the heartbeat evoked potential. They cleverly acquired ECG and EEG data and manipulated attention and found that if you attended to the heartbeat, then you modulated the amplitude of your evoked potential, this showing a conscious correlate of interoceptive attention. So we have two of the authors, Kao and Katerina, here, so congratulations to you both. It was a very tight vote at the end. The Editor's Choice Award of the Year, toward defining deep brain stimulation targets in MNI space, a subcortical atlas based on multimodal MRI histology and structural connectivity. This is a beautiful paper, which is in many ways NeuroImage. Multimodal tissue maps, highly quality controlled segmentation, introduction of a histological atlas, and tractography is added. And all of this is performed in a sophisticated Bayesian platform, and this yields a composite atlas, which is of course of high importance to the field at the moment, with the emergence of invasive deep brain stimulation technology. We liked it because it addresses an important topic. Advanced multimodal data fusion is popular in many different applications. The attendance to single subject customization and the fact that the atlas is publicly available. So I'd like to invite the senior author, Andreas Horn, representing Sean Ewart and the other authors.
Thank you so much. We're really honored, Siobhan and I, and I'm really happy to accept the award on her behalf. She can't be here, unfortunately, but she's really thrilled, as everybody who knows her can imagine. And I wanted to briefly mention that the paper was actually a long birth, so it received the reject and resubmit votum twice, which is probably a record, but the reviews really helped us make this a great paper. So if you reviewed that paper, you're part of this award as well. And what I also wanted to mention briefly is we visited, so it was a year process from the first preprint to the final paper. And during that time, we visited the lab of Malar Chakravarti, who's also here in Montreal, who contributed the histology in that paper. And I think that really boosted the overall paper, so I wouldn't be standing here without Malar today. And the final thing I wanted to say is that the paper was part of a special issue led by my personal heroes, a young team of editors, and I really, so there was Pierre Belec, Cameron Craddock, and Brian Evans, so it was a segmentation special issue, and they were really supportive in the whole process, and we're really happy, so thank you once more for this award.
Good morning. My name is Christophe Michel. I'm the editor-in-chief of Brain Topography, and it's my pleasure to present the Best Publication Award of Brain Topography, sponsored by Springer. The editors of Brain Topography award this year's prize in honor and memory of Professor Fernando Lopes da Silva, who passed away the 7th of May. Fernando Lopes da Silva was one of the most influential neurophysiologists of our time. He pioneered many EEG and MEG analysis methods and shaped our field like no other. The editors of Brain Topography would like to honor his long-time support for the journal. The paper that we awarded for the 2018 Best Publication Award was selected, was a review paper entitled Visualizing the Human Subcortex Using Ultra-High Field Magnetic Resonance Imaging by Max Keuken, Bethany Isaacs, Robert Trampel, Witzke van der Zwaag, and Birte Forstmann from the Universities of Amsterdam, Leiden, Maastricht, and the Max Planck Institute in Leipzig in Germany. The authors made a very comprehensive literature overview of studies devoted to the imaging of subcortical structures using high-field MRI. 169 publications are summarized and discussed in great detail, and the advantages and the limitations of different imaging sequences for the different substructures are discussed and illustrated in this paper. So the main conclusions that they made out of this survey of all this literature is that the subcortex can be parceled into a large number of anatomically distinct structures, and that only approximately 7% of these known structures are currently incorporated in standard anatomical MR atlases. They also conclude that ultra-high field MRI with a wide range of imaging approaches has been able to identify 169 subcortical structures in this individual brain, which should be incorporated in future atlases. So given that the different tissues, that's what they found, require different MR sequences and parameters, it is important to experimentally determine the optimal sequence for each structure of interest. So we, the editors and chief editors of Brain Topography, consider this review a great reference for the emerging field of ultra-high field MRI, and also for the given the increasing interest of the functional and the functional significance of these different subcortical structures. So I'm happy to hand over the prize to the first co-author, Bethany Isaacs.
Thank you. I'd just like to say thank you to the editors. My co-authors and I are really excited to have received this award. And also thank you to all of my colleagues at the Forsman Laboratory in Amsterdam.
Good morning, and we are going to start the LOC Symposium, Back to the Future, the Legacy of Italian Neuroscientists for the Current and Brain Mapping Challenges. I would like to remind you that today there is the post office for the special cancellation for the OHBM celebration, and today and tomorrow. So take advantage of this to have your souvenir. I welcome also Danielle Perani, who is my co-chair this morning. And we are among the, let's say, the earliest neuroimagers in Italy. The symposium, the LOC Symposium, is supposed to give a sort of flavor of a country which has the honor to host the OHBM meeting that year, and with their scientists giving a speech. Speeches about what they are doing and why that's relevant for research in neuroimaging and where that comes from. So it's my pleasure to invite the first speaker, Professor Francesco Pavone from Florence, who will talk about from Golgi's histology to current monitoring of brain rewiring. Please, Professor Pavone. 15 minutes, 15 minuti. Sì, c'è.
So, good morning, everyone, and it's my great pleasure to be here. And I'd like to talk about the fact that that optics, it's a great, let's say, opportunity to go behind the resolution that we have usually in MRI or standard techniques. But one of the main issues is the large volume. So I'm going to treat today how optics is going to treat about big volumes. And in that perspectives, how does it work to go on here?
Con il mouse.
Yeah, but does it work? So You know, the brain has particular features, and I mean, when you're going to do imaging and you really want to dissect, let's say, images from the nanometric scale, what you want to see is in the same image having both the dendrite and single spine image, and then to place that kind of image in a context of a dendritic arboring, for example, and then place that dendritic arboring with the long-term connections in the place of the whole brain. This is a mouse brain, for example. So the big challenge from the, let's say, imaging point of view, it's the optical dynamic range. That's to say that now I'm able with my iPhone to make a photo to you, okay? And then zoom in, maybe the one is already sleeping, and then zoom in every single cell of that person with the same image. So it's the capability to zoom in with the same image. That means having a sensors which is huge. It's a terabyte, it's petabyte sensors on which I can store a single image, and then I can choose every kind of details that I want with a submicron resolution, which is about 1 billion times higher than MRI resolution. But the brain has also other challenges, is that I have to label the brain in a way that I'm able to make a multi-level imaging, multi-level mappings from fiber connection to neurotransmitters, to cell distribution, molecular maps, genetic expression, and then the dream would be to overlap this structural functional, they say structural connectivity with the functional connectivity, and having both on the same volume. One way to do that in the optics is to use a sort of computer-assisted tomography scheme, which is exactly like CAT, but using photons instead of radiations. And that allows us to have an enormously higher, let's say, sensitivity and enormously higher resolutions. In this way, the sample, which can be 1 centimeters cube sample, of course we are talking of ex vivo imaging of fixed samples, can be placed in an objectives, illuminated transversely and observably orthogonally by a detector, by, let's say, in this case, an objectives. In that way, for example, the sheet of light, it's called light sheet microscopy, can be moved in all the direction. The sample can be turned around, moved around, and then at the end I can have the 3D tomography of the brain. But one of the main problems is that if I want to look a big piece of brain, let's say 1 centimeters cube, for example, this is not transparent, this is absorbing, and so I won't be able to see it. So the trick here is to make the brain transparent. This is obtained with a mouse brain, but later I will show you on the human brain the same results. And the trick to make the brain transparent is to remove the water, because the water is an index of refraction different from the extracellular matrix. And if I substitute the water with a fluid index matching, I can have an homogeneous index of refraction medium, and then it's transparent. Then in this case, if the mouse is a transgenic mouse, and for example neurons are labeled with GFP, I can have a look to the neurons, for example. Or I can, for example, in this case, reconstruct completely the entire brain with a resolution of a single spine. This is a mouse brain, this is cerebellum, for example. This is the low resolution image. I can zoom in inside and see single spines, I can see axons, and so on. But then an interesting thing is I can perfuse a gel into the mouse, and then I can reconstruct the entire vasculature of the brain mouse. In this case, this is a low resolution image. It's a 30 terabyte image. Then of course I can zoom in and having the high resolution where every single capillary is visible. And that's very interesting because I mean, I can use some, let's say, numerical simulation to simulate MRI signal in this way, but we will talk about that later. Then I can place everything in the MRI system. So I can do, before sacrificing the mouse, the same vasculature imaging with an MRI, and I can superimpose the two images. So I can place the optics, let's say, volume in the MRI volume, and but with a completely different resolution. For example, the light sheet microscopy, it's 1 micron cube. The MRI resolution is 100 micron cube. So in the volume is 10 to the 4th times the voxel bigger. So I can have a lot of information, much more with optics more than MRI. And then, of course, I can take a transgenic mouse, I can superimpose the two images, the vasculature and the neurons, and then I can have a sort of neurovascular coupling. Of course, here I need to label other cells like glia and other kind of cells, but this is what is totally doable. So by using and by learning this stuff, we learn in the Human Brain Project, which is the human the flagship, the European flagship about, let's say, the mouse and the brain simulation anyway. We moved to the NIH initiatives, and then we were asked to map the human brain optically. So this is the brain of a volunteer from France. It's an old woman that donated her brain. And of course, at that point, we, instead of having 1 centimeter cubes, we may have slices of brain, slices of 10 per 10 centimeters, and the thickness can be 1 millimeter, can be half a millimeter. And then these slices can be put on this scanner. Here we have a double SPEAM light sheet microscope, which is absolutely very, very fast. It scans about 0.1 millimeters cube per second, so an entire human brain can be scanned in 3 months, which is very, very fast. Of course, here we have a problem with data, but I will deal about that later. Of course, then we have two things to be done differently from the mouse. First of all, we have to clarify the human brain. This is doable. It's another protocol, which is called switch. We recently developed a clarifying medium application with TDE, which works very, very well. But the problem here is that we cannot have, let's say, transgenic humans. So we have to develop some post-mortem labeling. Switch, it's a protocol that enables to have the brain by removing the lipids like a hydrogel, and is able to label with antibody 1 millimeters thickness slice of 10 per 10 centimeters, okay? And in a quite reasonable way. What you can see down are some images done at different depth. This is the labeling of parvalbumin in this case. And this is an image of a stack of human brain, a slice of human brain, where we have DAPI labeling from neurons and parvalbumin with antibodies as well. But then, of course, we try more than 100 antibodies in the NIH project we are working on, but at the moment, 20% are working. But still enough, I would say, to have a quite high number of colors in our mapping. And you can see here main, let's say, labeling antibodies that we are using, and some kind of images. And this is, for example, the entire hippocampus labeled with the NUN and GAD67. This is, of course, the low-resolution image. You see the squares. This is not an artifact. This is every, let's say, area of imaging of the microscope is 1 square. Then we have to move with the scanner the piece of tissue to the next area. Of course, this is something that you see here that we can process, and we can make the image totally uniform, but this was not done just to show you how many images we have to stitch all together to make just a slice of an hippocampus. And of course, a slice is 1 millimeter. So in Z, we have many planes because we have a resolution of 1 micron in Z. So we have 1,000 planes in Z. This is a maximum intensity projection. Low resolution, of course, because we cannot show a high resolution image with such a let's say, huge image. But if you zoom in, you can see every kind of detail. In fact, you can zoom in. This is the cortex, for example, labeled with NUN and DAPI, and you can see here every neuron very, very well. The problem is that you want to extract information. So you must use artificial intelligence or deep learning to segment neuron. And you have to do in real time, because you are collecting such a huge number of data that you cannot wait for post-processing. So here we develop a new deep learning algorithms, which is able to segment in 3D the images. So we have a new kind of microscope, a sort of, let's say, virtual microscope, because instead of having an output, a raw image, we have a vectorial image. We have the already segmented images. And this is very important also for data science issues. This is the raw image on the left. This is the segmented vectorial image on the right, and this is done in real time. So you don't need post-processing. It's very, very fast. And then at that point, here we go. The points that you have are the neurons. So we image a big slice of a brain, and immediately we have the neurons segmented in their shapes. You see, of course, here points, because the image is enormous, but if you zoom in, you see exactly the neurons that you've seen before. And this can be done on the different slices, these different pieces of the brain, as you can see here. This is, for example, the slice of half a millimeter, you can see here, where this is a synthetic image, where every point is the reconstruction, a 3D reconstruction of a neural body. And then, of course, we can use the antibody to label a class of neurons, and then to dissect the subclass of neurons by segmentation, by recognizing the shape of the neuron. So this morphochemistry approach allows us to have a very eye-specific, let's say, recognition of neurons. And here on the right, in blue, in different colors, you have different kind of neurons labeled with the same antibody. And so the cell census now, it's possible in the big pieces of brain, and soon in the whole brain. But very interesting, by a serendipity effect, we recognize that during the process of bleaching of the brain, we increased a lot the self-fluorescence of myelin. So for free, without labeling, we had all the myelin fibers labeled. Self-fluorescence, with a very huge fluorescence. And this works on mouse, on rat, on monkey, and on human. And so at the moment now, we have the possibility to reconstruct not the entire population of neurons of the brain, but also to reconstruct in high resolution also all the myelinated fibers. And that works very well. On mouse, this is the low resolution image. This is the high resolution image of a small insect. The same for rat. The same for velvet. The same for human. This is a piece of human hippocampus, for example. And it's very interesting. For example, for rats, you can see very, very well some pathologies that alters the myelin, let's say, organizations. This is a normal mouse on your left, and the real mouse, the realer mouse hippocampus. And if you zoom in, you can see immediately by eye the difference in myelinated structures, organizations of fibers. And by using some very simple algorithms which recognize the spatial frequency, the organization, and the space, you can very, very well recognize the differences. Also very interesting when you compare this with DTI images. The DTI images from MRI, they can show fibers as well, but with a much, much lower resolution. And one of the problems of DTI, when two fibers are entering the same voxel, they are not recognizable anymore. Now we can compare DTI with this kind of system. We can train again DTI to increase by reverse engineering the fiber tractography with much higher resolution. And then at that point, you can put everything together. You can put fibers and cells. This is a piece of hippocampus. You can see, for example, here, all neurons with n are labeled, and myelinated fibers are autofluorescent, so you can have the whole structure all together. Of course, the problem is big data. And to do better, we need to make bigger volumes, we have better resolution, we have to be very fast, and we have to acquire as much channel as possible. And that's the big challenge It's not only optics, it's computer science, it's deep learning, it's artificial intelligence applied to imaging. And for example, if you compare one human brain, for example, if you compare one human brain with 3 channels, for example, with 1 micron resolution it's 9 petabyte. And then if you want, of course, to collect 9 channels, it's 27 petabyte, only one brain. And then if you have hundreds, you have thousands and thousands of petabytes. So at that point, of course, what is really needed now is to reduce the dimension of memory. What can we do for that? Otherwise, we don't have computer on the heart which is able to analyze such a huge amount of data. Consider that, for example, the memory storage for detecting only one human brain is the same of the USA academic research libraries. So, one possibility, of course, the way to do is to pass from the real images, raw images, to vectorialized images, which are segmented images. Here, for example, you have all the vasculature structure of the mouse brain. So when you segment a mouse brain, and then you have another image, which is 1,000 times lower in terms of capacity of memory, but has the same information. And it works very well. For example, here you see superimposed the real image with the segmented one, and you see in red the area where the segmented and the, let's say, and the raw image are not overlapped. It's less than 10%. So it works very well. The challenge here is segmentation on 1 petabyte images. And this is 1 centimeter image. Now we're moving to 1,000 centimeters cube image, and that's the real challenge we're facing in this moment. But still, 1 centimeters cube of tissue, it's about 6,500,000 planes, because every image of a microscope, it's a few hundred microns square, 1 micron thickness. Then 1 centimeters cubes, it's 6,500,000 planes. And you have to stitch all those planes all together to make a 3D images. So we developed recently a deep learning algorithms, which is doing automatically, because you cannot do manually, of course, which is 20 times faster with respect to state of the art. And now we are able to stitch in a 3D volume of 1 centimeter cubes in less than 2 hours. Then, of course, you have the problem that the images are enormous. So you can compress. We are working with a team that developed the MPEG protocol. You know, the MPEG protocol is the protocol that you have on your mobile for looking movies. And they ask us to use that protocol in neuroscience. And that, I have to say, works very well. We arrived to have a compression of about 700, okay, without losing any data. Let me specify what does it mean, losing any data. It means that if the quality of image is the same, but when we segment image, we don't lose any data. I mean, the space in which we don't have to lose any data is the vectoral space, not the raw space. So now, if we have 1,000 of reduction moving from the vectorial to the raw, from the raw to the vectorial, and 700, we have about 700,000 of compression. So now we can move from a supercomputer to a laptop computer. Then, of course, you have to extract data. And to extract data, it is not only, for example, recognize the volume and the shape of neurons, as I told you before, as I showed you before, but we have to recognize also where neurons are into the brain. And of course, if you take a huge image of a lot of centimeters cubes, hundreds of centimeters cubes, it doesn't mean that in every point the quality of the image is the same. So you need, again, another artificial intelligence tool to make a sort of semantic deconvolution. In other words, adapt the thresholding to recognize neurons point by point in the brain. And by using neural networks, now it is possible. And for example, in 1 centimeters cube, it is very possible to recognize all the neurons. We did for mouse. And what you see here, it's a synthetic image. It's the cerebellum of a mouse, where every point is a position x, y, z into the space of every single neuron. And we can count all the cells with a precision of about 95%, a recall of about 97%. So, let me close with some provocative messages. Of course, mapping the human brain is absolutely interesting, is absolutely important. It helps us, also to develop a new, not only to understand the brain, but to develop new pharmaceutical targets. But the next, let's say, frontiers, it's not only having the super high resolution structural connectivity, but being able to superimpose the structural connectivity to functional connectivity. So the donors that the brain that we have from such donations, it was without any, let's say, functional data. It would be nice in the future to have people that they donate their brains, but donate also their signals. So if we have functional data with structural data, then we can really have at that point, because we can move from the MRI to the optics very fast, we can really have a good superposition of functional connectivity with structural connectivity. But also reconstructing, let me say, is a provocative message. The, let's say, the vasculature with such a high, let's say, resolution, by modeling, by using mathematics, we can somehow model the F-bolt signal. And so at that point, if we are able to model the F-bolt signal, model the F-bolt signal, we can use reverse engineering and use the F-bolt signal to detect very high resolution, much higher than usual clinical data of the vasculature or of the signals. Let me also say that neuromorphic computing would be inspired by that. And one of the main challenges of neuromorphic computing is that there are much less energy consumption. In other words, if I'm going to use a supercomputer that is simulating my computation of my brain's consumption is about 20 watts, like a low energy consumption. But if I use a supercomputer, it's 10 to the 9 watts. It's about 10 to the 8 times higher. So having a neuromorphic computing which learns absolutely from the brain is very important. But also the new class of artificial intelligence. Now we are talking a lot of artificial intelligence, but artificial intelligence is very artificial because it's not inspired by the brain. But the new frontier of artificial intelligence is the artificial natural inspired intelligence. And that's, it's doable only by knowing very well the structure of the brain, both in its structural and in its functional characteristics. And knowing that, we can at that point learn from the brain how the brain is going to manage distributed networks, which are the neurons, but tomorrow they could be power grids, e-health networks, infomobility, security, or even drug discovery. And say that, let me also thank you for your attention. Let me thank all the people from the group at Lens, but also the people participating in this project from Sinai University, Mount Sinai, Boss from Boston University, Fischl from MGH in Boston, Peng from Seattle, Micera from Geneva, Mattia and Rofili from Bioethics, which are developing the artificial intelligence tools, Christophe Dextre from Tours, Valerio Conti Guerini from Mayer Pediatric Hospital, and finally Francesco Orsini Frasconi from the University of Florence. Let me also thank you for your kind attention.
Thank you, Professor Favone. Thank you.
We have to go on.
Yeah, we do not have time for questions as we have to finish by noon, and I remind every speaker to stay within 15 minutes, please. Daniela, please.
Okay, we have to go on because the time is running, so it's my pleasure to introduce Maurizio Corbetta from Padua. He will present us a lecture titled From Muscles Waiting to Representational Function of Spontaneous Brain Activity. Thank you.
Thank you. Good morning, everybody. I think we have to go back. This is right in the middle of my talk. Can you put the first slide, please? No, no, dietro, dietro. Dietro, dietro. Stiamo andando avanti. We have to go at the beginning. You have to slide back to the first slide, please.
Potete mettere la prima diapositiva, per favore?
Okay, thank you. Okay, well, thank you everybody. And Francesco, that was fantastic. I took the advice of the LOC to try to give you a little bit of history about Italian neuroscience. So my talk would be a little different. I'll tell you first about Angelo Mosso, what did he do for a few slides. And then I will discuss one experiment that will try to test the idea that the functional connectivity, the spontaneous activity patterns in the brain, are actually not just related to structural connectivity or neuronal synchronization, but are actually representing information states. So, Angelo Mosso was born in Turin in 1946. He graduated from medicine, also in Turin, summa cum laude. Then he went to Lipsia to learn how to measure vascular fluctuation using different kinds of devices. He came back to Turin and eventually established as a professor of physiology the Institute of Physiology that became the best academic institution at that time to study the brain. Mosso was also kind of a Renaissance man. He was interested in archaeology. He's also set up a lab in the Alps to study the effects of hypoxia. That was in 1907. And eventually, he also apparently was also a bit of a rascal because he died of syphilis in 1910 from secondary syphilis. Now, he was of humble origin, and his father was a blacksmith. And he was able to learn how to use his hands and build devices. And in 2014, in the Accademia dei Lincei, which is the Italian National Academy of Science, this new manuscript of Mosso surfaced. And there was a nice paper by Sandrone and collaborator, and Daniela Perani, our co-chair, is on the paper. And in this paper is described very beautifully the first neuroimaging device that Mosso designed. This is a balance table, very precise balance in which the subject was on the table. And then at that point, with a lot of controls, the subject was asked to perform different kinds of cognitive operations. And depending on the tilt of the table, Mosso tried to determine whether increase in blood flow or increase in circulation due to cognitive function would eventually tilt the table towards the head. Now, this experiment was then replicated in London. Unfortunately, the results are mixed. We still don't know whether your brain changes its weight when you're performing cognitive functions. Now, I did a little bit of research in my own institution in Padova. Padova is an old university. It's been around for about 800 years. So we have an old institute, and we have a very damp and dark basement in which we have all the historical books. So I went digging out in the library, and I discovered this other book by Mosso, The Temperature of the Brain, La Temperatura del Cervello, in which now he's using thermometers, mercury thermometers, very precise, that he was actually to place into the brain of these people with open skull injuries. This is Luigi Cane, one of his patients. He was a worker, a brick kind of fell on his head, creating a fracture. He also had a CSF leak. And Mosso was able to insert the thermometer inside the parieto-occipital region of Luigi Cane and ask him to perform different kinds of calculation. In another experiment, he used something called a plethysmograph, which is basically a machine to measure pressure in void. And so he was able to apply, let's see if I can use here my mouse. Mouse doesn't work, but anyway, here on the right side, you see the cup. The cup was applied over the parieto-occipital region, and then the little cuff here indicated by N and M, that was applied on the foot. Vacuum was produced in both devices, and then Mosso was able to record curves of fluctuation of the pressure in the brain, on the surface of the brain, that's the tracing on the top, and then he was able to also measure fluctuation in the fluctuation of the blood vessel at the bottom. And what you can see in the top trace, you see this beautiful slow frequency fluctuation from the brain. And then at some point, he asked the subjects to perform a multiplication, 21 times 13. And now these workers start, as you can see, the fluctuation from the brain. There is an increase in volume, a slight increase in volume. And you also see that the amplitude of those fluctuation is increasing. At the point W, towards the end of the tracing, you see that the subject now is performing a response. The response was actually accurate, and you see even a greater fluctuation in the brain. Simultaneously, the peripheral blood vessel, this seems to shrink. There seems to have a decrease in volume, and you see also decrease in the fluctuation of the peripheral capillary. Probably the subject were very much stressed out, his noradrenaline went up, and that probably caused a vasoconstriction of his peripheral bed. Now, as you read the book, it is pretty clear that Mosso was concerned with some of the same issues that we're dealing with in neuroimaging. He knew that he was doing measurements from the brain using indirect means, which were very low signal to noise, not different from fMRI. He was also using multimodal measurements to try to get some grip on the sources of the variability. On the table, on the balance table, he had posts that was measuring actually head motion, and he was also had belts to measure respiratory artifacts. He also used modern task design, comparing rest to task, and also comparing parametric manipulation of cognition. One thing that I really like about Mosso's experiment is that he was actually to measure simultaneously both the brain and the body. I think this is really one of the next frontiers. We've been studying the brain as almost like a disembodied organ, the brain by itself, but we really need to understand how the brain and the body are connected. Now, one of the issues that I think is fundamental in neuroscience today and that links the brain and the body is the question, what is the function of brain energy? The brain weighs only 2% of the body weight, yet it consumes about 20% of the body energy, and we still don't have a clue what is all that body energy for. There have been a lot of calculation, theoretical and empirical calculation, trying to understand where the energy is going. We know that both in the cerebral cortex and in the cerebellar cortex, about 1/4 to 1/3 of the energy goes in maintaining the membranes, changing the mitochondria, basically kind of protein production, mitochondrial leak, and other non-neuronal function. However, 3/4 of the energy goes in neuronal signaling, of which action potential that are energetically very expensive consumes very little, maybe between 10 to 20%. And most of the energy goes in synaptic transmission, postsynaptic transmission, that is slight membrane oscillation at the postsynaptic side, whose function is at this point not very clear. At least in the cerebellum, Atwell and collaborators have done calculation, tried to compare different computational strategies in the cerebellum. And they've argued, based on this metabolic calculation, that the output of the cerebellum, the Purkinje cells, are actually using only 20% of the energy. When the cerebellum kicks in and is basically correcting or anticipating the motor response in the motor cortex, that is what the 20% is about. However, 80% of the energy in the cerebellum is in mapping all the possible input and output operations, all the possible repertoire of stimulus response mapping that the cerebellum can do is actually where the energy seems to be consumed. So at least in the cerebellum, we don't know in cortex, but at least in the cerebellum, the energy seems to be going to representing stimulus response or stimulus cognitive response mapping. And that leads us to the question of the resting state. So this is a movie of a brain, a rest. These are the fluctuations that we're measuring with fMRI. And these are some of the ideas that people have put forward over the last few years. One idea is that this functional fMRI fluctuation are reflecting synaptic homeostasis. In other words, is maintaining the synapses connected. And this explains why structural and functional connectivity are similar to each other, but particularly under conditions in which there is less cognition, for instance, sleep or coma. Another idea is that these fluctuations are reflecting communication through coherence, neural synchronization between regions that are activated during a task, and then through Hebbian plasticity, this activation, this correlation are then maintained in the resting state. A third hypothesis that what I'm going to test with my experiment is this idea that the brain is maintaining representation in the intrinsic activity. In other words, it's representing not only the communication between regions, but it's actually representing information states that are important probably for biasing what we might be doing during cognitive tasks. So the hypothesis then that many people have proposed, and our group has also worked on this, is this bilateral relationship between task-driven activity, determined to some extent by genetics, development, and your own experience. So our brain, although very similar, is slightly different in every one of us. And this task-driven activity can then lead to fluctuation in the resting state. And in turn, this fluctuation in the resting state can be considered spatiotemporal priors for task-driven activity. What I'm concerned with, however, is the multivariate representation in every particular brain region and the changes between these regions in these multivariate patterns. This is what I'm looking at. Typically, regional or IC-based functional connectivity is considering a region as a unit, as a node. We talk about node and edges, but of course in any regions there are like hundreds or thousands of voxels. So we're going to test this idea in human visual cortex. Human visual cortex is a system in which you have a gradient of activity as shown by people like Nancy Ken-Wisher and Jim Axby. They're basically a gradient that represent different kinds of information: faces, bodies, tools, scenes. And one of the most interesting results of this line of work that has been going on now for 20 years or more is that there is a low-dimensional structure in human visual cortex. There is basically a difference between the activity pattern for animate stimuli, faces, bodies, mammals, and non-animate stimuli like tools, chair, musical instruments, and scenes. And this very low-dimensional structure, you can see it here in human IT cortex. So what we're interested in asking is, are natural visual stimuli represented in human IT cortex also during spontaneous activity? And in order to do this, we developed a data set of stimuli containing 10 different categories and multiple repetition of the same stimulus or different stimuli for each category. And first we scan people with a block design to identify a region in human cortex. Then we scan people with an event-related design to basically get a multivariate classification for each individual exemplar and then for a category as a whole. The experiment went like this. We first got resting state, so the subjects did not know what they would be doing in the following of the experiment. This resting state was acquired for about 30 to an hour in different sessions. Then we obtained localizer scans to localize regions in human occipital cortex. And then we obtained event-related scans to basically get multivariate classification. So first we localize the region in extrastriate visual cortex, and we see the classic regions that everybody has been studying. EBA, by the contrast of body versus objects, fusiform face area, face versus objects, and parahippocampal place area for scene versus objects. Of course, these are the canonical regions. In reality, we have multiple regions for each category. So we have 6 or 7 body region, 3 or 4 face regions. We have about 9 to 10 scene regions. And so for improving the classification and simplify the computation, we put everything in a joint ROI. That is, we vectorize these regions and we call it a joint body ROI, face ROI, and scene ROI. Then we test the representational similarity, the kind of experiment that Jim Axby and others have done. And now we present multiple objects. For instance, here at the bottom, you see multiple multivariate responses for different kinds of scenes. We can then look at the representational similarity across all the stimuli. And you can see that in EBA, for instance, stimuli that are classified as mammals, body, and faces produce more similar responses in task evoke activity than other kinds of categories. We then take all the body regions and we obtain a body joint ROI, in which we also get representational similarity maps. And now this is basically a map across all the body regions. The fourth step is taking this template of different categories or individual template, individual stimuli, and slide them along the resting state of the subjects before the experiment was performed. So here you see on the left side the multivariate template for the scene average template. And in the resting state, you see that curve that represents the similarity between the stimulus pattern and the resting state pattern frame by frame. And on some frame, you get, for instance, on the right, in the purple, a very good match between the stimulus and the resting distribution of activity. In other frames, for instance, in the green frame, you get no match. And in the blue frame, now you get a negative match. You get both transient, negative, and positive matches, and a lot of non-zero matches. So we can draw now a distribution of correlation values, and we're going to take the U90 value, the top 90% values, as our measure of basically match between the resting activity and the task activity. And at this point, what we can do, we can put all this distribution and compare the distribution with control stimuli. In this case, the control stimuli are the phase scramble phases. These are artificial stimuli that, according to the two-way reciprocal relationship between task and rest, should not be represented as commonly. And what you can see in the distribution, you see that the target category phases are actually wider, so that you have basically more matches for phases than artificial phases. And at the bottom, you see the U90 values, and you see that again in the resting state, you get a better similarity at rest for the categories that are related to phases and not similarity for the other categories. We repeat the comparison for body, and we get exactly the same results, and again for scenes, and we get again the same results, and now you can see that the scene similarity in the scene preferred ROI is actually the highest. So at this point, what I show you is that at rest, there are moments in time in which your resting state activity is resembling canonical pattern of task evoke activity within a region. Now the question that I would like to ask is whether this is actually co-occurring in time so that we have phase state-like patterns in visual cortex as compared, say, to body-like patterns in visual cortex across multiple regions. And in order to do that, I'm plotting here the 6 different body regions in terms of their similarity between task and rest. And you see that although, of course, the average has a lower similarity value, there is quite a bit of variability, but all of them show kind of a peak at the level of the body category. And over here, we see the scene regions. And again, there is a little bit of variability, but all of them, they show a match at the moment in which the scenes are presented. At this point, we're ready to look at the functional connectivity between these regions. So if we take 2 body regions as an example, and 2 scene regions as an example, and now we're taking the body pattern, and we're sliding the body pattern across the 2 body regions, you see at the bottom, the blue and the azure line, that those regions are showing correlated fluctuation in body template. If I go in the scene region, now I see that when I show a scene template, those regions will show fluctuated correlation in scene template, but not in the opposite one. And what you can see here is that these states are actually occurring independently. At this frequency, either you have a scene template in your brain or you have a body template in your brain. When you look at all the regions, and across all the different subjects, this is, we get a nice, at the bottom, double dissociation. So when you use a body pattern, body ROI will show more similarity at rest. When you use a scene pattern, now scene regions will show a greater similarity at rest. Now what about the correlation with the functional connectivity? When we go and look at compare what we call pattern-based FC, which is basically reflecting this multivariate classification with ROI-based functional connectivity, we see in every subject a match between 0.7 and 0.85, suggesting that what we measure as functional connectivity is actually a combination of pattern states that are cycling through in time. So, to conclude, fluctuation of resting activity in visual cortex encode preferred stimulus patterns more frequently than artificial stimuli. This intrinsic pattern fluctuation respects the topographical selectivity of visual cortex and the categorical similarity. So, in FFA, face and body patterns are more likely to occur than scene patterns. Now, you might ask why we see a change in the variance and we don't see a change in the mean. We don't know why that is, but one explanation is that we're only testing 40 faces, 40 bodies, 40 scenes. We're not testing the millions of faces you might get exposed to. But nonetheless, these results has also been shown in animal work. These are data from Amir and Greenwald, optical imaging in monkey cortex. They developed task classification task classification for orientation stimuli, and now they slide the stimuli in the resting state. And you can see here that the mean also in their distribution is about 0, with peak positive and negative peaks in primary visual cortex during spontaneous activity. Okay, so I'm done. Thank you very much. And I'd like to thank my collaborator, Do-Young Kim and Gordon Schulman. And the paper now is in review. And some of you reviewers, please be kind with this paper because we think it's very important. Thank you very much.
Thank you. Thank you, Maurizio. We are sort of very late on time, so I'm pleased to invite Professor Maria Concetta Morrone from Pisa University. She will be talking imaging the visual system from Gennari and Santorini to brain imaging. Grazie. 15 minutes strict, please.
Okay, thanks very much. Thanks for the invitation. Can I have the first slide? La prima diapositiva, per favore. Okay. Thanks very much. Thanks for the invitation. It's a pleasure to present today the legacy, the Italian legacy for the visual imaging of the brain. And we have to go back to the 1700s, where a great anatomist like Domenici Santorini that studied in Pisa, and you'll see Pisa will be an important center for visual science. He, for the first time, perhaps, is the first demonstration that I know that really show that experience can change, can mold your brain. Indeed, he observed something that now Pietro is an expert of it, Pietro and Emiliano. He observed that acoustic nerve is much more prominent in people that are blind. And this is really something that still we don't understand the mechanism. Perhaps it's the myelin, we don't know. But perhaps the most famous Italian people that everybody knows is Gennari, another great anatomist that indeed we call the primary visual cortex, triad cortex, because Gennari discovered this incredible, it's very hard for me to point, but doesn't matter, this incredible stria that is the input of the optic radiation from the thalamus, lateral genicular nucleus to the cortex in layer 4. And now we know a lot of the organization of layer 4. We know that this layer 4 receives independent input from the left and right eye, and this input goes and builds what is called now ocular dominance, mapping or ocular dominance column. Now, where we know a lot, and a lot to the contribution to our knowledge about the organization, microscopic organization of the neural pattern in V1, are derived from the vision laboratory in Pisa, led and started by Lamberto Maffei and Adriana Fiorentini in the 70s. And I think that the greater contribution of this lab was also to link architecture, neural firing with function, how we see. And that is really what most of us does with functional imaging, really the link between function and local organization. And we know also, going back to Santorini, that this ocular dominance column can change during the critical period. And indeed, experience mold our brain. But we can change dramatically this territory, this cortical territory dedicated to eye, mainly during the critical period. And later on in life, we think that we cannot change. So this has been a great dogma for many, many years. However, it will be important to reopen the critical period later in life because we can in this way promote self-repair of the brain, and we do need to learn how to do so. So I think one of the last contributions of Lamberto, very important, was really to show that he can reopen critical period in the rat by pharmacological mean, but is a rat. What about human? And again, another great contribution comes from Adriana. We just lost recently Adriana, which she showed, perhaps it was the first time, that the first demonstration that indeed the human brain late in life too can, the primary visual cortex, can be reorganized plastically through perceptual learning. Yeah, but what about ocular dominance? This is perceptual learning, it's very local circuitry. Can we also change ocular dominance in adult? And here I move to what we are doing now in the PISA Vision Lab. And this was a question that we asked a few years ago, but instead then go and measure and track what has been always considered Hebbian plasticity, the fact that if you don't use for a long time one input, the cortex will suppress that input. We went and studied the other side of the coin that we call homeostatic plasticity, that is really related to the fact that after short-term deprivation, the brain high up is getting the gain up of the deprived input. So you patch one eye, and what is happening? Well, you first measure ocular dominance perceptually, usually it's the vision between the two eyes balanced. Then you patch one eye for 2.5 hours, and what is happening? That input, that vision becomes much stronger. Is prevailing over the vision of the other eye by a factor of 2 and more. Well, but what this does means, do we have exactly the same mechanism that we have during development? So we studied the mechanism underlying this phenomenon using ultra-high resolution field that we needed to do because we had only 10 minutes because the effect is not permanent, it's decaying very fast. And we used both we measured GABA, that is one of the implicated molecules during the development, the one that seems helping closing the critical period during development, in a scanner with Claudia, Emir, and Olli in Oxford, and more recent in a bold experiment in Pisa with Michela, Paola, Jan, and Laura. So let's see first the GABAergic or the data on GABA. So we measure GABA before deprivation, after 2.5 deprivation, in a voxel that was positioned on the occipital cortex, and we found the result only on those voxels, not in any other part of the brain, like parietal cortex. And you can see this is a GABA of one subject before deprivation. This is GABA measured in that voxel after deprivation. And you see that is decreased. Well, it's one subject. Well, we repeated in 20 subjects. And you see there is a clear decrease of GABA after 2.5 deprivation in the resting GABA, so with eyes closed. But what we think is more important, because it's a linking function to imaging, is that the people that had, in the subject in which we measured the strong decrease of GABA, are also the subject that showed the stronger effect of homeostatic plasticity, the one in which the boost of the deprived eye was stronger. That is GABA, let's see the bold. It's also this boost associated with a local increase of activity. So this is the data, the experiment that we just published, that we presented in these 10 minutes that we had before deprivation, a series, sorry, it's not going, yes, a series of stimuli, low and high spatial frequency. Here I'm reporting the mapping of the response in along V1, V2, V3, and V4. This is the stimulation of the deprived eye before deprivation, and this is after deprivation. Notice by eye that here the response is larger. If we go and see the non-deprived eye, the response is decreasing. So the eye that has prevailing vision has also higher bold activity, particularly strong in V1. So V1 changes. So when we go and do all the analysis and ANOVA, we found that the effect is highly significant, but again, what is again important, people that did not have a strong functional effect also did not have this boost of the eye special frequency in V1, and the correlation is quite good. So this correlation really suggests that there is a strong link between GABA and bold manipulation by change after deprivation with the function. But this is V1. If you are interested to this story, go and please and see the poster that Paola will present tomorrow, where we see this effect even in LGN. And this is again is another large, big dogma that rely plasticity just at cortical circuitry. Okay, but you should tell me, well, why you are so much interested in this short-term transient plasticity. I mean, who cares? You cannot really change the brain in 10 minutes. Well, perhaps we have means, both pharmacological, but also more physiological means in which we can stabilize that plasticity over time. And we approach this question using a particular type of pathology that is amblyopia, that is lazy eye. Probably, 5% of you will have a lazy eye, or at least if you have not been cured during development. So what we did, to make permanent or try to make more long-lasting this effect, what we did, a counterintuitive therapy, we patched the amblyopic eye, we put the subject on a bicycle and watching a movie. So we combine physical exercise with deprivation. We repeated the training just 6 times, every time for 2 hours. So overall, 12 hours of training. And this is what are the results. The visual acuity of the amblyopic eye dramatically decreased, improved during the training, nearly by a factor 2 line. You said, well, 2 line, but for these people, 2 line meant a lot. And what is important, the association with the physical exercise really stabilized this homeostatic plasticity, so the effect was present after 1 month, 3 months, and after 12 months, a year. Importantly, some of these patients, not all, also become somewhere really stereo-blind, and they recover some stereoscopic vision. So just in conclusion, and I am in time, I hope, visual cortex retain a high degree of plasticity in adult. Homeostatic plasticity induced by short-term monocular deprivation is associated with change in cortical excitability, a decrease of GABA, a boost of neural gain, phenomena that we do see during development in animal model. Physical exercise can be a physiological means to stabilize and to promote plasticity, and perhaps going on along this way, we can find also other strategies to endorse plasticity in other illness to promote self-repair. And I would like to conclude, remember I was Lamberto Lab many, many years ago in the 80s, and thank all the people in our lab in Pisa, the Pisa Vision Lab. Thank you.
Thank you, Concetta, very nice talk, and for being on time.
So I have to call Alessandro Bertolino. Of course, he will have the proper time needed for the presentation, which are 15 minutes, and we have to ask the next symposium 3 more minutes to be spared for our. So the title is Beyond Lombroso, Genes, Imaging and Environment in Mental Disorders.
Thank you very much, Daniela. Thank you very much, Pietro, for inviting me here. It's an honor to be here to give this presentation in such a prestigious hall and to represent Italian neuroscience here at this prestigious meeting. So you've heard the talk, and the title of the talk, and let me briefly introduce you to who Lombroso was. He was a physician who was also a criminologist, and he insisted that he had a theory of anthropological criminology, which essentially stated that criminality was inherited, and that someone born criminal could be identified by physical defects which confirmed a criminal as a savage. So this physical information that he was trying to gather from which to understand whether a person was anthropologically criminal was especially some facial features. Of course, there isn't much to be proud about the theory of Lombroso in the sense that it gave birth or it participated to some of the eugenics that followed afterward in the following century. But what it was very clear to him, it was that there were some human behaviors that are heritable or inherited. And following on this line of thinking, what we have been studying in my laboratory in the past few years are these two main lines of research. One is to try and understand the genetic and environmental risk for schizophrenia, and the other is how can we identify some biological pathways which are prone to gather genetic risk for schizophrenia. So let's talk about the first line of research first. As many of you know, many psychiatric disorders are largely heritable, and these disorders include schizophrenia. This is information treated by a review paper by Sullivan, and you see that disorders like schizophrenia, autism spectrum disorders, and bipolar disorder are highly heritable, meaning that around 80% of the variants of the phenotype is explained by genetic variation. But genetics is not everything for many common disorders. There are also environmental risk factors which are recognized to be risk factors for schizophrenia, for example. One of these risk factors is obstetric complications, and another one, which has also been recently established, is being victimized, being bullied by other people during adolescence. And in the search for schizophrenia genes, up until a few years ago, several labs in the world concentrated on the so-called hypothesis-driven genes, or single gene approaches, and our lab focused on the dopaminergic pathways, especially the dopamine D2 pathway, which has been implicated in the pathophysiology of schizophrenia for decades based on a treatment with antipsychotics, which are dopamine D2 blockers. And then this landmark paper came out a few years ago now. This is the first wave of the second wave, actually, of the Psychiatric Genomics Consortium, which gathered a large number of subjects and performed the so-called genome-wide association study. And this was the first study that unequivocally established that there are 108 genetic loci associated with diagnosis of schizophrenia. Now, without going into much details about this, time is limited. A few of the key points of this paper were that many of the genes involved by this study were not genes that we could have known before, and only a few of them actually made sense from the neuroscience that we knew before, like, for example, the dopamine receptor D2. But most of the genetic variation that has been associated with diagnosis of schizophrenia by this study is intergenic, and it seems to serve a regulatory purpose in the translation of DNA information into mRNA and protein afterwards. But one of the also important points that this paper scored is that now we can have genetic information about each single individual, in which we can calculate a polygenic risk score once we know all the genes, or at least many of the genes that are involved in genetic risk for schizophrenia, based on the allelic information present in each individual person. We can calculate for a single person a polygenic risk score, and with this polygenic risk score we can summarize what the genetic risk of that single individual is for schizophrenia. What is the advantage of this? first advantage and most obvious advantage is that we can calculate the interaction with environmental risk factors. And this is what Danny Weinberger and Gianluca Orsini did in this paper published in Nature Medicine, in which we also collaborated, in which the following was demonstrated. First of all, as you can see on the graph on the left, of course, the polygenic risk score, which again summarizes the genetic risk for schizophrenia for each individual, of course, as you would expect, is greater in patients with schizophrenia than in healthy subjects. But when dividing up the subjects based on whether they had undergone early life complications like obstetric complications, you can see that the result changed quite significantly, in the sense that between patients and controls with no early life complications, there is no significant difference, whereas there is a statistical interaction between genetic risk for schizophrenia, diagnosis, and early life complications, in the sense that patients with schizophrenia have greater polygenic risk score in the presence of early life complications, suggesting an interaction between an environmental risk factor, which was already known, and the polygenic risk score in patients with schizophrenia. And of course, the greater is the early life complications, as summarized with this score here, the greater you see the difference between patients and healthy controls. Also, with the polygenic risk score, we have summarized the whole genomic risk for schizophrenia. But what are the genes that do provide an interaction with the early life complications like obstetric complications? And we can actually see it in this graph. A placental polygenic risk score was calculated versus a non-placental polygenic risk score. Basically, a polygenic risk score based on genes expressed in placenta versus genes not expressed in placenta. And you see that the bulk of the significant difference is found in the polygenic risk score for which the genes are expressed in placenta. So suggesting that it is the placenta mediating this interaction between obstetric complication and genetic risk score for schizophrenia. And ontogenetic analysis also showed that response to stress and genes responding to hypoxia are the genes that are involved in this complication, in this interaction. Also, in our lab, Giulio Pergola also performed another study between polygenic risk score for schizophrenia and another known risk factor, environmental risk factor, which is being victimized and being bullied. Giulio opened up a collaboration with a group in the Netherlands, which had studied a group of around 600 kids, followed up longitudinally over 3 age periods of time: 10 to 12 years old, 12 to 14-year-old, and 14 to 16 years old. Just remind you that the peak of age at which there is greater risk for schizophrenia and symptoms start to appear is between 14 and 16 years of age. And what Giulio has shown here is that genetic risk for schizophrenia interacts with being bullied by other people only when at a specific age range, which is between 12 and 14 years old, not before and not after, and that this interaction, as evaluated with a mediation analysis between genetic risk for schizophrenia, indirectly predicts the frequency of psychotic experiences at 16 years of age, and the mediation effect is performed by bullying victimization, suggesting again a specific way of how genomic risk interacts with environmental stress factors. So now that I've shown you a few examples of genetic and environmental risk for schizophrenia and how they interact, we can see how we can try and use different kinds of information to understand how genomic risk for schizophrenia may involve different biological pathways of risk for schizophrenia. So we have used the following approach. We have used the transcriptome-wide information from post-mortem brains in collaboration with the Lieber Institute for Brain Development, for example, in order to study the whole transcriptome, the whole human transcriptome. And we want to, again, here the attempt is to try and characterize biological pathways which are involved by genomic risk for schizophrenia. So in order to categorize the information in the whole transcriptome of the human brain, we use co-expression analysis with this software which is called WGCNA, Whole Genome Co-Expression Network Analysis, because the idea, one basic tenet of biology is that genes are co-expressed together. Large groups of genes are co-expressed together because they are co-regulated, namely identifying specific biological pathways by means of how these genes are co-regulated and co-expressed together. So by analyzing co-expression analysis, we hoped and we think we have succeeded in identifying specific biological pathways which are completely data-driven. No hypothesis is involved in this. Of course, once we identify this network information, this co-expression information, we want to try and translate this genomic risk for schizophrenia into in vivo information, because we want to understand what's happening in vivo with the belief and the hypothesis that the genomic information is shows up in different phenotypes that then lead to the phenotype of schizophrenia. These different phenotypes, for example, being functional brain imaging, with which we study brain function in patients with schizophrenia, we have been doing this for the past 30 years, so we know that there is some differences between patients and controls. So how do we bring this information from post-mortem to in vivo phenotypes? By using a polygenic co-expression index, we use a very complicated way to calculate this thing, but basically what we do is that we characterize the genetic variation, predicting the co-expression of a set of genes as analyzed with postmortem. So with that number, the polygenic co-expression index, we can perform associations and correlations with in vivo imaging phenotypes or other phenotypes. And the imaging phenotype we have been working for the past 30 years is working memory cortical inefficiency, which we and others around the world have demonstrated is a main characteristic of how the brain in patients with schizophrenia processes information in an inefficient way. And from animal studies of Patricia Goldman-Rakich, for example, it has been shown clearly that the dopamine D1 and dopamine D2 receptor contribute significantly to determine efficiency of brain processing, especially during working memory. So this is the first example. This is just a graph showing what we did, which is summarized in these two papers here by Giulio Pergola and Pierluigi Selvaggi, published a couple of years ago. And in this first study, we performed the method, we established the method, and then what we did, we characterized a set of genes which is co-expressed with the dopamine D2 receptor from a microarray study of the dorsolateral prefrontal cortex from post-mortem human brain. We calculated the polygenic co-expression index, and what we did then, we used the polygenic co-expression index to perform the association with human brain activity in vivo, of course, in healthy subjects. You see, we obtained two independent replications. This is a sample from the Lieber Institute for Brain Development again. So two independent, one independent replication of our results, showing that the greater the polygenic co-expression index, namely the greater the co-expression of the whole set of genes being co-expressed with the dopamine D2 receptor, the greater the activity of the dorsolateral prefrontal cortex during performance of the NBAC working memory task, which is consistent with what has been shown with PET studies and fMRI studies before by the group of Slifstin and others a few years back. Also, recently we also published another paper in PNAS in which Leonardo Fazio, Giulio Pergola, and Giuseppe Blasi from my lab performed a similar study studying the set of genes being co-expressed with the dopamine D1 receptor. And again, we have a discovery and replication analysis in which, again, there is a similar relationship between the polygenic co-expression index of the dopamine D1 set of genes with cortical activity during the back working memory task. And you see that the direction of the correlation this time is negative, suggesting that the more co-expressed are these genes, the more efficient is the cortical activity during working memory. Finally, paper which is coming out in biological psychiatry. What the next step, of course, we wanted to try and evaluate is whether any of these sets of genes as identified with network analysis is enriched for genetic risk for schizophrenia. Long story short, Giulio and Pasquale Di Carlo identified a set of genes which is highly enriched of genes that are known to be involved in risk for schizophrenia. For those who know something about the genetics of schizophrenia, you see that several of these genes have long been known to be involved in genetic risk for schizophrenia, like CACNA1C, GRM5. So once we identify this module, which is enriched for schizophrenia genes, and this is actually replicated across 5 different samples, we want to see whether there is any relationship of this module of co-expressed genes, so a biological pathway which we, in a data-driven fashion, we have identified, whether there is any correlation with, for example, something which is very important to any psychiatrist in the world, which is response to antipsychotics, in this case from the Katie trial and from our own sample, independent replication again, in which we show that there is a significant correlation, that the higher is the co-expression of these genes, the lower is the response to the antipsychotic treatment. I think I've been very on time. Just some acknowledgements before quick conclusions. I would like to acknowledge the collaborators at the Liber Institute for Brain Development, like Luca Orsini, Anand Mattei, and Daniel Weinberger, who also happens to be my mentor, and then Fabio Sambataro at the University of Udine, and the PIs in my group, like Giulio Pergola, Giuseppe Blasi, Grazia Caforio, Antonio Rampino, and Linda Antonucci. Just 1 second for very simple and plain conclusions. Identification of genetic variants associated with schizophrenia is ongoing, so we are at a good stage with this, but we need to understand how this genetic risk, these genomic loci create biological risk for schizophrenia. The interaction between genetic risk and environment is something that we need to keep into account. And we believe we are using a tool like gene networks, genetic networks, to identify biological pathways which translate risk for genomic risk for schizophrenia into in vivo phenotypes, which we can also measure. Thank you for your attention.
Thank you, Alessandro. And